Let us have a look at this question. What does it say? Where is the middle lamella found in a cell? So we are talking about middle lamella. Now first important point, where exactly is middle lamella present and what exactly is it made up of? Middle lamella is actually, keeping it very simple, is the cementing material between two plant cells. Alright, what is middle lamella? It is the cementing material, right, present between two plant cells. Alright, so when you look at this image, right, when you look at this image, what you see here are these blue colored spaces. Right, now that exactly is what is middle lamella. And to keep it even further simple, you must have definitely seen in your life a wall made up of bricks. Right, when you see the bricks, what do you see in between them? You would be seeing that different bricks are attached and stuck onto each other like this with the help of cementing. Right, with the help of cement. Right, like this. Okay, so what you see as the shaded region, what I have drawn, and what you see as the blue colored thing in the given image is the cement kind of material, right? So this cementing material or this cement kind of holds the two things together. When we talk about brick, the actual cement holds the bricks together, and when we are talking about the plant cell, it's actually the calcium and magnesium pectate which are present as the predominant components of the middle lamella. Alright, so that actually forms the cementing material. And why exactly are we calling it cementing? I'm repeating it because it is technically allowing the cells to kind of stay together. It is holding the cells to be present together. Alright, yeah. So now let us have a look at the options. So where is the middle lamella found in a cell? Is it inner to the primary cell wall? Is it inner to the secondary cell wall? Is it outer to secondary cell wall? Or is it present to outer to the primary cell wall? Okay, so when you look at a plant cell, one of the first layers of the cell wall that is formed is the primary cell wall. Alright, let me write it down here. So primary cell wall. This one is the primary cell wall. Whenever the secondary cell wall is being formed, it will form inner to the primary cell wall. So this one, inner layer, becomes the secondary cell wall. Alright. Now, if I'm making a cell adjacent to this, I'm making a smaller one. Yeah, I'm making another cell which is adjacent to this. Where exactly will be middle lamella present? It's going to be present outer to the primary cell wall, like you see in this image. Alright, so it is present outer to the primary cell wall. This green colored thing here is primary cell wall. Okay, now do you know when is it formed? When is middle lamella formed? One of the first depositions which starts happening for the formation of cell wall during the cell division process is actually the deposition of the calcium and magnesium pectates. Okay, so when the cytokinesis or the cell division starts to happen for a plant cell, the calcium and magnesium pectates starts laying down and they start forming the wall, that is the cell wall of the daughter cells. Alright, and this is what is termed as the cementing material. So middle lamella, which lies outer to the primary cell wall, is termed as the cementing material for the plant cells. Let me summarize everything for you all. The right answer here would be outer to the primary cell wall, as I already specified. The middle lamella is the layer that is first formed during cytokinesis stage, which is division of cytoplasm. Cyto is referring to the word cell, right? Kinesis means movement. It's because of the movement of the cells apart, right? This happens during the cell division, and the additional layer that is formed after that is the primary cell wall. So when you talk about just the layers of the cell wall, the first layer to be formed is going to be primary cell wall and then secondary. And when you talk about overall cell division process, like what is the first stage of formation of cell wall, that's going to be the formation of middle lamella. I hope that point, that differentiating point is clear to you all. Once again, when you talk about just the cell wall layers, the first wall to be formed in the young growing cells is going to be primary cell wall. But when you talk about cell division also, like when the cell is coming from the parent cell. It's the first deposition is of the material of the middle lamella, is of calcium and magnesium pectates, right? Hence, the middle lamella is found outside the primary cell wall. The primary wall is the first layer of cell wall and is mainly made up of cellulose and hemicellulose in plants. What are these? These are basically the polysaccharides which are forming structural component of the cell wall. It is formed inner to the middle lamella and is typically found in young cells. Like I just specified just a while back that young cells, whenever it is growing, the predominant layer there is the primary cell wall. After that, when the cell has attained certain level of growth and whenever maturation happens, that is when the secondary cell wall starts coming into picture. This then gets either replaced or the secondary cell wall is going to be added inside towards the cell membrane, that is inner to the primary cell wall, right? So the right answer for the given question here is option D, that is outer to the primary cell wall. Let us have a look at this question. What does it say? The cell wall shows. We are talking about the cell wall, and when you look at the option, we are talking about the permeability. Is it impermeable? Is it semi-permeable? Is it differentially permeable? Or is it freely permeable? Alright, now first thing is, what is the meaning of permeability? Whenever a membrane or a partition or a wall that we are talking about here, it allows the molecule to pass through from one side to the other, we are going to term it as permeable. Alright, based on the degree of permeability, whether it is allowing all the molecules to pass through, whether it is allowing only the solvent molecule or molecules up to a certain weight, right, atomic size, it is allowing them to pass through, we are going to call it as semi-permeable or selectively permeable or differentially permeable for that matter, right, based on the degree of permeability. And whenever a membrane is not permeable to something, we call it as impermeable. Alright, when do we call a membrane as freely permeable? When it basically allows all the kind of molecules, irrespective of their chemical nature, to pass through it. But the major point here that you have to remember is that even though it will allow all the kind of molecules to pass through it, it will still have an exclusion principle based on the size. Because just imagine, if a molecule has to pass through a membrane or a wall, it at least has to be of a smaller size than the pore size of the membrane or the wall, correct? So here only the principle of size is going to come, but beyond a certain size of that particular molecule, the cell wall or the wall would not allow those molecules to enter inside. Alright, according to the properties that a cell wall has, a cell wall is completely permeable in nature. Obviously, it does not allow molecules beyond a particular size to kind of enter through it, to kind of pass through it based on the pore size of it, right? And this weight actually is nearly about 30 to 60 kilodaltons, alright? So, let me summarize everything for you for this question with a little more theory. So, the answer that you have got already is freely permeable or free permeability, correct? The cell wall is a freely permeable layer, and it is freely permeable to small molecules, but as I specified, there is a size exclusion which is 30 to 60 kilodaltons. Now students, some of you might not know what exactly is Dalton. Dalton is basically equivalent to atomic mass unit, alright. It is equivalent to atomic mass unit. What is an atomic mass unit? It is basically 1/12 of the weight of a neutral carbon atom, alright. Now this is something that you already know. Okay, Dalton is just a new term for you all to introduce or for you all to learn. Please remember, most of the molecules, when we talk about biology, it is measured in terms of Daltons or kilodaltons. Alright, I hope that makes it clear to you. So here, molecules which are bigger than 30 to 60 kilodalton would not be able to enter the cell wall. But otherwise, cell wall is kind of a freely permeable membrane. An impermeable layer. What is an impermeable layer? Impermeable is not going to allow things to pass through it. One of the best examples for this to be understood is example of a cork, right? You have seen cork stoppers which kind of stop the bottle or the mouth of the bottle. Now why is cork used in this kind of a way? It's simply because cork has all the cells which are highly deposited with something called as suberin. Okay, what is suberin? It is a lipophilic substance. It is a lipid loving substance, and hence it kind of is almost impermeable to water, because of which it does not allow anything to leak out of the bottle or to enter through the bottle. And that is the reason why we say cells which have a lot of suberin are one of the best examples of impermeable cells, right? Completely impermeable layer. So an impermeable layer does not allow any molecules to pass through it. You've understood an example of impermeable also. The question is asking for cell wall, but it is good for you to remember at least one example of all kind of membranes or walls or layers, right? Talking about a semi-permeable membrane or differentially permeable membrane, one of the best examples for this is our plasma membrane. Okay. In fact, plasma membrane is not just semi-permeable or differentially permeable, it is highly selectively permeable. What is the difference between semi-permeable and selectively permeable as such? Semi-permeable usually allows all the solvent molecules to pass through it. Selectively permeable membrane is going to allow the solvent molecule, yes, but it also allow certain different kinds of solute molecules based on the specific properties of the membrane and the molecules to pass through it. Alright, so selectively permeable becomes even more specific of a membrane, and that is what is the property of our plasma membrane is. Cell membrane and all the membranes of the organelles are said to be semi-permeable, and plasma membrane is specifically speaking selectively permeable. I hope you all got this question correct. The right answer here is option D, which is free permeability. Let us see what this question is asking. Members of the eukaryotes are by nature. So we are talking about eukaryotic members, or rather eukaryotic organisms. They are by nature unicellular, multicellular, acellular, or both A and B. Okay. Now what are eukaryotes? Eukaryotes are those organisms which have a proper membrane bound nucleus, right? So this is first property that we look at when we talk about eukaryotic cell. Membrane bound proper nucleus which is organized with all histone proteins, etc. And the second major point of difference that comes for the difference between prokaryotic and eukaryotic cells is the presence of membrane bound, I am writing it down in short, cell organelles. Okay, like you talk about endoplasmic reticulum, you talk about mitochondria, you can talk about Golgi apparatus, lysosome, etc. So here in eukaryotes, the membrane bound organelles are present as well as the nucleus is present, which is a membrane bound structure which has the DNA wrapped around, or rather organized with the histone proteins. Now, one of the major points that you have to remember here is that even though eukaryotes are kind of higher organism, they may still be unicellular, and I am sure that you remember one of the best examples like euglena, paramecium, amoeba. These are the examples that you've been learning from your junior classes, right? So all these examples that I just listed are all eukaryotic cells, but they are all unicellular, right? They are all unicellular. So eukaryotic cell can be either unicellular or it can be multicellular. Like for example, look at ourselves. Look at yourself, right? Look at the animals around. What are all these organisms that I'm talking about made up of? They are made up of multiple cells. They are made up of various organ systems which are forming because of various different organs which are formed in turn with various lot of tissues, and each tissue is actually nothing but a group of cells which are performing similar functions, right? So this is a multicellular organization. All right. So when you look at the options here, it is obviously going to be option D, that is both A and B. Let me show you quite a few relevant images here. What you see here is the image of a euglena. You see how euglena is moving? Yeah, it is basically a single cell. It's eukaryotic, but it is a unicellular eukaryote. When you look at rabbit here, yeah, a cute little rabbit, this is multicellular in organization as I specified. And when you talk about multicellular organisms, how does the organization take place? Like I specified, there are cells which are organized into tissues. tissues are basically in turn nothing but a group of cells which are performing similar function and are similar in structure. Then different types of tissues combine or come together to form different organs. Like you talk about your stomach is going to have muscular tissue, it's going to have connective tissue, it's going to have your nervous tissue as well, it will have the epithelial layer also. So all these things are various different types of tissues which are forming a particular organ. When you talk about organ system, talk about your digestive system. It starts with your mouth, tongue, teeth, goes into the food pipe, into the stomach, into the duodenum, and then to the intestine, large intestine, and to the anus. So what is this? This whole thing is called as digestive system. So each system like this, there is going to be excretory system, respiratory system, circulatory system. Each system is made up of various different organs, and these organ system, that is nervous, endocrine, your digestive, your circulatory system, your respiratory system, your excretory system, they all come together to form a multicellular organism, which is the eukaryote organism that we are talking about here. So all in all, the right answer to the given question is option D, that is both A and B. Members of eukaryotes are by nature, they can be both unicellular or multicellular. Let us have a look at this question. What does it say? Cytoplasm is the jelly-like semi-fluid matrix found inside a cell. Alright, that is a fact that we know. It is composed of which of the following? Water, cell organelles, micromolecules and macromolecules, or all of the above. Now, before we actually get into the details of the option, let us first understand what exactly is cytoplasm. As it is already given in the statement of the question, what is cytoplasm? It is the jelly-like, very important, semi-fluid, right? It's basically, because it is jelly-like, we are calling it a semi-fluid matrix which is found inside the cell. Alright, what is so important about cytoplasm is that cytoplasm consists of almost 70% of water, with all the other organic and inorganic compounds suspended in it, along with the cell organelles present there. Cytoplasm basically helps in filling up the whole cell, and it holds the regular organelles that are present in the given cell. Be it a prokaryotic cell or a eukaryotic cell, it holds all the organelles in the cell at their respective places. And also, because of the fluid that is present in the cytoplasm, it allows all the metabolic activities to occur in itself, right? So very important point is, because metabolic activities are all happening in the cytoplasm and the organelles which are present in the cytoplasm, it basically is consisting all the micromolecules and macromolecules as well. Okay, micromolecules are what? Smaller sized biomolecules, and macromolecules are going to be the bigger ones, right? So all these micromolecules, macromolecules, 70% water like I already specified, and the cell organelles are all present in the cytoplasm of a given cell, right? When you look here in this image, this is a very simple representative image of a eukaryotic cell, where you are seeing all the membrane bound organelles being shown here, right? All these organelles that you are seeing here, starting from endoplasmic reticulum going up till the Golgi apparatus. There is mitochondria that you see here. You see the centriolar apparatus that is present here. You see Golgi like I was specifying here. There is nucleus, membrane bound nucleus that is present here. So all these cell organelles are present in this given eukaryotic cell. But also when you talk about the prokaryotic cell, membrane bound cell organelles are not there. Ribosomes is there, the nuclear DNA is there. They are all suspended in the cytoplasm itself, along with of course having a water background, as well as along with I was specifying inclusion bodies in case of prokaryotic cells. So all these things are all present in cytoplasm, which allows the dynamic state of the cell to really function. So the right answer here is going to be option D, that is all of the above. Let me summarize everything for you all. Answer is as I specified, all of the above. In both prokaryotic and the eukaryotic cell, a semi-fluid matrix called cytoplasm occupies the whole volume of the cell. This is the main arena of cellular activities in both plant cell, animal cells, as well as prokaryotic cells. Now, plant and animal are eukaryotic cells, but it is valid for prokaryotic cells as well. Various chemical reactions occur in it to keep the cell in a living state. That is all the metabolic activities we are talking about. It is mostly made up of water, and other micro and macromolecules. Also, various cell organelles are present within the cytoplasm. I hope you all got this question correct. Let us have a look at this question, and this is a very important question. An elaborate network of filamentous proteinaceous structure, we are talking about an elaborate network of filamentous proteinaceous structures which help in the maintenance of cell shape is called. Okay, the options are cytoplasm, cytoskeleton, endoplasmic reticulum, and then Golgi complex. Okay, let me give you a quick background first. Now, when we talk about the maintenance of the cell shape, cytoplasm also helps in doing that, right? Because it is kind of the semi-fluid matrix of the cell. It holds all the organelles in the place, etc. But very important point, the question is not asking just the structure which helps in maintenance of the shape of the cell, but it is also asking about the filamentous and proteinaceous structure. Now, cytoplasm is not solely protein. It is made up of a lot of micromolecules, lot of macromolecules. It is made up of a lot of amount of water, etc. Okay, so we cannot say that cytoplasm is the only thing that is helping in the maintenance of the cell shape. All right, so this cannot be the right answer. Okay, cytoplasm is a rejected option here. Then we talk about let me talk about endoplasmic reticulum and Golgi network first. Talking about endoplasmic reticulum, which actually starts immediately from the nuclear membrane, from extension of the nuclear membrane, and it goes up till the start of the Golgi apparatus, and then Golgi apparatus kind of forms a membranous structure till towards the end of the cell. Okay, slightly away from the cell membrane, but yes, lysosomes and all, there are continuous apparatus when we talk about the endoplasmic reticulum and Golgi apparatus and lysosomal systems. But once again, the point here was about filamentous proteinaceous structure. Okay, so that is the trick here, and that is why I was specifying that this question is very important from understanding, from conceptual point of view and from exam point of view. Okay, you all got the point that I am trying to convey here. A cell is maintained with its shape with multiple factors, but the question here is specifying about the proteinaceous and the filamentous structures which are helping in the maintenance of the cell shape. So the right answer here is going to be cytoskeleton, option B. Okay, let me tell you about each one of the options here. First thing is let me tell you about the right option here first, which is the cytoskeletal apparatus or cytoskeleton. What is cytoskeleton? As the name is specifying, let me write it down here. Cytoskeleton. If you break the word cell, skeleton. So cytoskeleton is nothing but simply speaking skeleton of the cell. Like how we have our internal framework where we are having an axial and an appendicular skeletal system. That is, you know, we have all the bones that are holding our whole body together, making it stand erect, you know, sitting posture, lying down, having the whole control, giving the whole framework to the body. In a similar way, cell has these beautiful proteinaceous filaments which are present across the cell in order to maintain its shape. This cytoskeletal apparatus or the cytoskeleton has three major types of cytoskeletal apparatus. Alright, they are microtubules, intermediate filaments and microfilaments like you see here. Okay, we have just taken a small section of the cell to show it to you, like how you see here. Microtubules are the thickest of the cytoskeleton, like you see already. You know, try to focus on the images that are given here. It is the thickest of the cytoskeleton, which is made up of tubulin protein mainly. Okay, it is made up of tubulin protein. So microtubule becomes the polymer of tubulin. All right, anything small which is building up a bigger unit is going to be called as a monomer, and anything bigger which is made up of repetitive units of the monomer is going to be called as a polymer. Okay, talking about intermediate filaments, actually one of the best examples of intermediate filaments is when you look at your hair, you all know that it is made up of keratin. So that is why it has so much of strength. Now can you imagine this to be present at a microscopic level? It is going to perform the similar function of giving so much strength to the cell. So these are the intermediate filaments. Talking about microfilaments, microfilaments are the thinnest of all the three, and they are made up of majorly actin proteins. What are they made up of? They are made up of actin proteins. Now these actin proteins, they literally work like simple twisting around of two strands. Okay, so actin are present in two layers and those two strands are simply kind of twisted around each other, and this twisting allows further strength to be present in the cell. Alright, I hope you all have understood the whole concept of cytoskeleton here. It is a very important concept from exam point of view, and this question, as I already specified, is equally important. Let me summarize everything. The right answer here, because it was asking about membranous filamentous structure which provides framework or shape to the cell, is cytoskeleton. Let us talk about the whole summary of the question. The cytoskeleton is supporting framework of microtubules, microfilaments and intermediate filaments, which are all proteinaceous: keratin, tubulin, actin, as the names are also ending with IN, which is something like an old rule of naming proteins, but of course it has changed now. So these are all proteinaceous in nature. This serves as the supporting structure for all the organelles found in the eukaryotic cell, because it's forming all the crisscross kind of network, so the organelles can be held in place. Talking about the incorrect options now, cytoplasm is the semi-fluid matrix which occupies the volume of the cell in both prokaryotes and eukaryotes. It is the main arena of cellular activities in both plant cells, animal cells, as well as prokaryotic cells. The various chemical reactions occur in it to keep the cell in the living state. We are referring to metabolic reactions here. Okay. Next, endoplasmic reticulum. I told you it's a network of membrane bound structures, but remember it's not just proteinaceous structures, right? So, network of membrane bound organelles extending from the outer membrane of the nucleus towards the plasma membrane. Talking about Golgi complex. By the way, what is the function of endoplasmic reticulum? Endoplasmic reticulum plays the function of lipid metabolism, protein synthesis, calcium storage, etc. And talking about Golgi, they are also membrane bound organelles which are pinched off from the endoplasmic reticulum, and they are called as the packaging organelle as well as the sorting organelle of the cell. I hope you all got this question correct.